at the bottom of your interactive notebook on page 44, you should see Wilkins and Franklin. That's where we left off. The last thing we talked about is Chargaff's, okay, where the different percentages of adenine equals thymine, okay. Now, today we're going to talk about some really big players in the history of DNA, okay. Wilkins and Franklin were two very important people. However, the people that we give credit to the structure of DNA, Watson and Crick, are the final people that we're going to talk about. So basically, our objectives are to know what these people had to do with the history of DNA. What part did they play? So, without anything further being said, okay, let's look at Wilkins and Franklin. Okay, Rosalind Franklin and Maurice Wilkins worked with the X-ray diffraction of the DNA molecule, and they tried to find more clues about the structure of DNA. So they were looking at the X-ray diffraction of the DNA molecule. Franklin's X-ray images suggested that there was a helical structure to the DNA. Okay? It suggested that there was a helical structure. Well, we know that DNA is a double helix, don't we? Okay? We know that the DNA is a double helix. When they took the X-ray diffraction, it looked as a double helix, a helical structure. Now, if you look, okay, here's Franklin, here's Wilkins. This little jobby in the middle, okay? If you see that, that is the X-ray diffraction. I gave you a picture in your notes that looks like it, okay? That is that very picture. That is the X-ray diffraction that they made. So they were looking at this, and they said, okay, it gives us the helical basis. Now, what happened later on with Watson and Crick, okay, with Watson and Crick was the big breaking point with the DNA, okay? So Watson and Crick were also working on the structure of DNA at the same time that Franklin and Wilkins were doing their work. Okay, so they're all trying to figure out the structure of the DNA. Now, Watson and Crick knew about Chargaff's rule that stated that A was always with T and C was always with G. So they knew that A and T were always together. They knew that C and G were always together. So they took Chargaff's rule and they were not entirely convinced about the helical structure that Franklin suggested. Okay, so they weren't really sure about this whole helix that Franklin was talking about. So that led her to, or that led her to doubt herself, her to think that, hey, maybe I'm not right. Well, okay, that's two gentlemen right there, okay. what happened is that Wilkins, who is not Franklin, right? Wilkins is definitely not Franklin. Without her knowledge, okay, without her knowledge, he went and consulted with Watson and Crick. He gave them the data that he and Franklin had worked on. He showed the X-ray diffraction to Watson and Crick. Okay? So he did not consult her, and he just took their stuff and went and showed it to Watson and Crick. Well, Watson immediately recognized the significance. Okay? They talked about the helical form. Well, when Watson saw that X-ray diffraction, he immediately knew that that was a double helix, because he'd seen a double helix before. So that went on to help Watson and Crick make their model of DNA. So could they have done, would Watson and Crick have found it? Maybe, but they would not have found it nearly as quick as what they did when Wilkins shared their data from their experimentation. Who's to say maybe someone else figures it out a lot quicker than Watson and Crick did had Wilkins not shared that information with them. Okay? So, now Watson and Crick came up with this whole idea that we have two outside strands that alternate with sugars and phosphates. That alternate with sugars and phosphates. If you look over here, here is a structure of a DNA, and it's not the twisted structure, but if you look, this yellow right here is a phosphate. This little ring right here, this 5 ring here, is deoxyribose sugar. Phosphate, sugar. Phosphate, sugar. Phosphate, sugar. Okay? Then they also went on to say, hey, cytosine and guanine paired together. Okay? Cytosine and guanine pair together, and you see these 3 little red dots there? Those 3-letter red dots signify hydrogen bonding. So there are 3 hydrogen bonds connecting the guanine and cytosine. Now, you know what at one time people thought? They thought that the purines and the pyrimidines were paired up. Well, what are the purines? AG, right? AG, adenine and guanine. Okay, look at them. They're both double rings. Okay, so those people that thought of the double rings, the purines being put together, their measurements were off. It didn't make any sense because, hey, you know, this doesn't measure correctly. Here's the pyrimidines only have 1 ring. So they said, but you know what? When we mix a purine with a pyrimidine, the measurements match up great. So that's when they determined that adenine and thymine. Okay, then they said, okay, the hydrogen bonds that hold them together. So right here are my rungs of my ladder. Here are the side rails. The side rails alternate sugars and phosphates. The rungs are either a thymine with an adenine or a guanine with a cytosine, but we have a purine mixed with a pyrimidine. So the measurements came out a lot better. Okay? So they said that thymine and adenine always pair together, cytosine and guanine. They got that from Chargaff's work. Okay? Now, they also went on to say that the strands are anti-parallel. Okay? And what they mean by that is that one strand runs one way and the other one runs the other. And they're talking about the 3' and the 5'. The 5' end is where the carbon is on the phosphate end. The 3' end, the carbon is on the sugar end. Okay, so look. Okay, 5' starts with the phosphate here. And the 3' ends with the sugar. See what I mean there? Okay, so the 3' end is the sugar end, the 5' end. Now notice how they are flopped. Let me get my little box out of the way here. Notice how they're flopped. How this end runs the opposite way, the 3 to 5'. So that's what the anti-parallel means, that the one way and the other way, they're going different ways from 3 to 5'. Okay, do you understand what the anti-parallel is? Now, if I look here then, I can go on a little bit more and say, okay, here, if you look at this, A goes with T, right? A and T go together, C and G. So now I've made my base pairs and I've matched my base pairs, but I also have in opposite 3 to 5' ends to be most correct, to show that actual base pairs are running in anti-parallel directions. Now, I just put this on there, okay? This is a medallion. I think it's a picture of the medallion from the Nobel Prize. So, Watson and Crick were awarded a Nobel Prize for the DNA double helix structure that they came up with. So they won a Nobel Prize for that. And if you look, okay, it's kind of hard to see, but they have ring stands and they have clamps and they have actually made up the double helical model of DNA. And they've made a life-size model that they said would be what the DNA actually looks like if you could see it under the microscope. Okay? So Watson and Crick have this DNA model that they came up, and for their knowledge on the DNA structure, they were awarded a Nobel Prize. Okay? Now, here's just some more things that I have on there. You don't really need, I don't think I have those. We've talked about purines and pyrimidines. Maybe we'll get to that a little bit more when we talk about RNA later